Benito, I have to say, I'm so grateful that I could carry on being a part of your explanations. All I want to do is contribute to your videos somehow.
Oh, that's alright, Sam. You know, I've always got your best interests at heart, you know. You can always count on me.
I know, Benito. I have to say thank you again, from the bottom of my heart.
Ah, yes. I mean, let's be honest here. I mean, quite frankly, you were never cut out for the job you had before.
There you go, Benito. Fabulous. Oh, thank you very much, Sam. Better go off to the video now. Right, here we are again. It's the 8th video of the sensory ecology series. We're halfway through, isn't that exciting? And today we're going to carry on looking at vision, but we're going to take a bit of a detour, to be quite honest with you. Because last time we talked all about rods and cones and photoreceptors and photoreceptive pigments. Today we're going to look at a branch of vision which doesn't actually require eyes or photopigments or any of that. Okay, and it's looking into a part of the electromagnetic spectrum which we can't see. Okay, and that's the infrared. So if we wanted to find out the thermal signature of the world, then we'd look at the infrared spectrum. And as I mentioned last time, on the electromagnetic spectrum, it's to the left of visible light. Okay, so it's at longer wavelengths. And we talked about with longer wavelengths, that means each photon of that wavelength will have less energy because E equals hc over lambda, so they're inversely proportional. Okay, so the less energy a photon has, the more difficult it is to detect, and the less likely it is to transmit through the atmosphere. So that means there's not much point in an animal being able to detect infrared at those wavelengths at all. Okay, so normally it's at the redder end of the infrared spectrum, if you know what I mean. Yeah, so it's around 800 nanometers, but then it extends to about tens of micrometers. Okay, so it goes quite a long way. In fact, there's not really a cut-off point before you go onto the microwaves, but you know, that's roughly where we are. So when a black body heats up, you know, things that are black because they absorb all the visible light wavelengths, right? Those black bodies will probably be emitting lots of infrared radiation, giving a thermal signature of the world. And as we talked about last time, not all light gets through the atmosphere. Microwaves, basically nothing. Completely the opposite end, the X-rays and the gamma rays, we basically get none of that. We get radio waves, obviously, and that's the really long wavelengths. But we don't know many animals that can detect radio waves. In fact, I don't think we know any. Okay, so that's just for our use, so we can listen to Radio 4 and all that. But infrared, a lot of it gets through the atmosphere. Okay, now the absorbance, there's this thing called absorbance coefficients of the molecules in the atmosphere. Because sunlight doesn't get through because it's absorbed by molecules, okay? And you've got your classic culprits here. You've got things like CO2, methane, water vapour, all those greenhouse gases which are brilliant at absorbing photons. Which is very good, by the way. We don't want to be receiving all that ionising radiation from the sun. So it's very good they do that, but you know, the greenhouse effect is a completely different kettle of fish. But the point is that some infrared does reach the Earth. So there is a window of opportunity, if you like, for animals to be able to perceive it and use it to their advantage. And, well, you've probably seen it on Spring Watch and things like that when they get their old thermal camera out, thermal imaging camera that basically sees heat. Okay, that's basically what happens. So if you like, we have this thermal zoo. Each animal has a particular thermal signature, which can be used by predators if they want to detect their prey. We'll look into a classic example of that in a second, but first we're going to look into something a bit different, okay? Beetles, fire beetles to be precise, and this was studied by Schmitz and Bonn, okay, in the 1960s. Now these beetles are able to detect forest fires, okay, from several kilometres away, something like 80 kilometres away I think, so huge distances. How are they able to detect that? Well, they're able to detect it through the heat that those forest fires produce. So of course these forest fires are emitting light in the infrared spectrum, around wavelengths of 2 to 3 micrometers, so quite long wavelengths really. Now we know already that the insect cuticle is brilliant at absorbing infrared radiation. Okay, because I mean the beetles are basically black after all, so brilliant. So this then allows coupling of the sensory organ to the type of energy available in that animal's sensory ecology, right? But first we need to get something straight. Why does a beetle want to detect a forest fire at all? Well, they're very cunning, these beetles. They know whenever there's a forest fire in places like Australia where it's really hot, then lots of stuff dies. So if a forest fire kills all the other animals off, that means a fire beetle who wants to lay eggs won't have any competitors, okay? They'll have no competition for resources or whatnot, because all the other animals that were once there have been killed off. So these fire beetles are actively searching for where forest fires are so they can lay their larvae on freshly burnt wood. They're what we call pioneer species. Okay? They're the first species to colonise a barren landscape and sort of regenerate the whole ecosystem. Okay? So they're the first ones to arrive. And lo and behold, an organ was found on the mesothorax of these beetles, okay? And it's got a dome-like structure and it's surrounded by glands. Here's a picture of it, yes? And these glands are to protect the detector from convective changes in heat. Now, there's something we've got to get straight here, because there's two types of heat. Obviously, we can detect heat, okay? If you accidentally put your hand on a boiling stove, then you're going to take it away pretty quickly, okay? So does this mean we have infrared perception? No. We can just sense temperature, okay? And that temperature is in the air. We can sense the heat that's in the air. That's basically due to the vibrations of particles, okay? Heat is basically all about the movements of particles, okay? How fast, how much kinetic energy they have, okay? Any Tom, Dick or echidna can do that, right? The other form of heat perception is what we've been talking about, this perception of infrared radiation, okay? So that's an important distinction then. The distinction between radiated heat detection, which is what these fire beetles can do, and convective heat detection, which most of us can do. And convective heat detection is rather short range, okay? So when you're able to detect a forest fire, which is 80 kilometers away, right, there must be something more to that. And this has now been demonstrated in 3 different genera of beetles. I'm going to look at another example later. So let's have a look at these receptors then. What do they actually look like? Well, here's a diagram, and as you can see, it looks a bit like an onion, doesn't it? And that's a good way of thinking about it, because the several layers of that dome receptor are crucial to how it actually works, okay? So as I've said, there's these concentric layers of cuticle. Now, when infrared radiation is absorbed, those successive layers expand, okay? Things expand when they get warmer, okay? Another basic law of physics there. So as those layers expand, then the shape of that dome gets deformed, okay? The shape changes. And that generates a force, okay? A different force on that dendritic tip of the sensory neuron, which you can see right at the bottom there, okay? So, if you get a force on that sensory neuron, then that has the potential, if it reaches a certain threshold, to fire action potentials, okay, which goes straight to the CNS, and the CNS can then work out what to do about it, okay? So if they've detected a forest fire, then they'll fly probably towards that forest fire. Now what's interesting here is that these sensory structures are very similar to structures which we see in hearing and mechanoreception. There's an organ, a sensory organ in our skin called the Pacinian corpuscle, which is a classic A-level example, I don't know, good memories of A-level, which is a very similar shape. It's like an onion, and when you put pressure on it, then it changes, it opens sodium channels and whatnot, and it fires off an impulse. And obviously it's very important to similar mechanoreception organs in insects as well. So this is what we call photomechanic conversion. So you could say that they're hearing the light, basically, which is a bit weird, isn't it? And actually, if you go into the genomics of this, we talked about last time about opsin genes. 7 out of the 11 opsin genes described in Drosophila, so a fruit fly, are expressed in the ears. So they're not just there, they're actually expressed in the ears of Drosophila. And obviously you can undertake behavioural experiments as well, where you insert an electrode at the base of the sensory neuron, and then you introduce some infrared radiation and measure the action potentials. And if you change the wavelengths of that infrared, then you'll realise that you'll get different thresholds for the firing of an action potential. It's interesting as well. And you can also do some intracellular readings as well, which involves sticking an electrode right inside the cell, so you could get some more accurate quantitative results on, ion flux and all that. Okay, so from one beetle to another, one which is very closely related as well. That last one was Melanophila. This one is Merimna, so different genera but in the same family of beetles, okay? This is Merimna latrata. And what's particularly interesting here is that these guys can sense infrared as well, but they use a completely different organ, okay? For a start, it's not even on the thorax anymore, it's on the abdomen. So let's see a diagram then of this organ. So when infrared radiation hits the cuticle, it activates these thermosensory cells, okay, which contain loads of microvilli and loads of mitochondria as well. That's one of the key features of them. Now, just like the Melanophila, thermal expansion is thought to play a role in the transduction process here. But the truth is, we don't really know, okay, the exact mechanisms behind it. So it's a bit of a mystery. So if you take neural measurements, then you see the usual. You get a refractory period, which is something you normally see if you deliver a constant stimulus, which is between action potentials. And then you can do similar behavioural experiments like what they did with Melanophila. You take neural readings by inserting electrodes and whatnot. You can even introduce a different form of light, a helium neon light, which emits light in the wavelengths of the visible spectrum, so around 600 nanometers, and see if there's a change in response. Because if there was, then that would indicate that these beetles are picking up on the infrared wavelengths and responding to them. Now, I've had loads of letters sent in saying, Benito, how are your Tenebria molotors doing? Simple enough question, I suppose. And seeing as they were talking about so many beetles, I thought I'd give them some limelight. They've had loads of babies. We've got about 30 Tenebria molotors in the house at the moment, and they are wonderful. Now, as far as we know, these can't sense infrared radiation. They're in a completely different family to the ones we've discussed so far. The ones we've discussed so far are in a group called the bark beetles. These are in a group called Tenebrionidae. I've just realised I just dropped one. This is now a beetle from a completely different family, Acanthocnemus nigricans. And this is a pyrophilous Australian beetle, so just like Melanophila. Pyrophilous means it's attracted to forest fires, okay? And here, yes, completely different sensory organ in a completely different part of the body again. So this animal uses two bilateral organs which are actually located on the head of the animal. So we've gone thorax, abdomen, head, the three main tagma of an insect, okay? So really going all over the place with this. So here's a diagram then. As you can see, it's kind of pan-shaped, and on that top membrane there, that's where all the sensory cells are. And if you take neural measurements, you'll realise that these organs have a high spontaneous rate and response inhibition. And this could be due to this internal cavity which you see below the sensory cells. Because that internal cavity lowers the thermal mass of that sensory organ. Now, what good can that do? Well, that could lead to a faster heat detection rate. Because if you have a low thermal mass, then you naturally heat up more quickly. Okay? So it's more sensitive, if you like, to changes in temperature, which is very useful. And it's also better at localizing that infrared radiation as well, if you've got a low thermal mass. So this decrease of thermal inertia allows for quick, fast responses, which allows infrared radiation to be detected quickly and localized quickly. Right. Now, the classic example which I was talking about earlier are the snakes. So completely different when we want to vertebrates. You've probably seen it on various, animal documentaries, things like Deadly 60 and whatnot, that loads of vipers in particular are able to sense infrared radiation. More specifically, they're using that to detect the heat given off by their prey, which are normally warm-blooded animals like rodents and things like that. And that's particularly useful if you're nocturnal, like many of the rattlesnakes, for example, are nocturnal, and you can't use your eyes very well. And they sense this infrared radiation by having pits, in pit organs, which are just to the side of the nose, below the eye, on a snake. Okay, they're little holes. And because you've got two of them, then perhaps these snakes can get some sort of parallax measurement to increase that localization of the prey. Okay, and you've probably seen it loads of times on television where snakes are presented with a hot balloon and it will pick that one over the cold balloon. Now what's interesting here is that the wavelengths that those snakes are detecting are of longer wavelengths now. That's 10 micrometers. They're the kind of wavelengths that are emitted from the bodies of small mammals. Okay, so these snakes are perfectly tuned in to what they need to detect. Now another interesting example which concerns us more is that of mosquitoes. Now if you've been in a tropical country, you'll know that, if you don't wear a mosquito net in the night, then you'll get bitten to buggery by mosquitoes, right? Now the mosquitoes can't see you. They could potentially be able to smell you a little bit, but it's been shown that they have what we call a sensillum styloconicum, which is able to detect the heat irradiated from your body. So these sensilla styloconica actually have a very sophisticated way of being able to detect temperature irradiated from warm-blooded animals quickly. Okay, so imagine a mosquito flies into a room and there I am, sleeping, naked, right? What the sensillum styloconica has, these warm and cold cells. So, I'm obviously warm-blooded. I've got a pretty cold sense of humour, but other than that, I'm a very warm person, believe it or not. And because of that, the warm cells in the sensilla styloconica will get activated. Okay, they'll produce action potentials willy-nilly. Yeah, they'll go crazy. The cold cells, on the other hand, they respond as well, but they decrease their firing rate. Okay, so both of them are firing normally, when there isn't anybody in the room, but when somebody is in the room or something warm-blooded, then the warm cells increase their action potentials and the cold cells decrease. So that quite clearly amplifies the signal then. It makes it more obvious. Okay? So the warm cells and the cold cells, you could say, are acting as antagonists. And obviously this is true for the other way around. So imagine if there's something cold in the room, then the cold cells will increase their firing of impulses and the rate of firing of the warm cells will go down. Okay, so it's very efficient. Now, a similar kind of system is found in ticks. Now, as you know, ticks are not related to mosquitoes at all. Mosquitoes are insects, ticks are arachnids related to spiders and whatnot. Okay, and this was studied by Gingle and Tiki, believe it or not, I don't know how you pronounce that, Tai Chi Tiki, something like that, in 2001. And these receptors are found on the front legs of the tick, so another completely different piece of anatomy for you. Now, the ticks can walk on those front legs, but when they get near a host, they tend to hold them up and, look round, if you like, to localise where that body is, okay? And they actually have two functions, because they actually are able to detect humidity in the air as well using those receptors. Now, we talked about earlier about the difference between radiant and convective heat. Now, locusts, there are no blood-sucking locusts out there. There's no reason whatsoever why a locust would want to detect, a warm-bodied animal, as far as we know. So we put that to the test. We exposed locusts, well I didn't, Gingle and Tiki did. What am I talking about? Gingy and Tiki exposed locusts to radiant infrared heat and convective heat. Now naturally, just like how me and you would, we would respond to convective heat. That's something we can all detect, yeah? We know when a room's cold and when it's not. But they didn't respond to the radiant heat. But the ticks did. Okay, that's the point here. That receptor on the ticks is what we call the coeloconic sensillum. And because it has two functions, because it can detect humidity as well, it's called a dual receiver as a result. But then again, heat and water are very closely correlated. If it's very humid, it tends to be very warm, because water vapour in the air, as we talked about earlier, the absorbance coefficients, it'll absorb more heat. Yeah, particles will vibrate and all that, so they're not completely mutually exclusive. Some types of receptors have also been thought to be able to detect CO2 as well, and that's certainly the case in the mosquitoes and the Styloconicum sensillum. Okay, so that's even more useful, because anything which emits CO2 could be something which you'd want to suck the blood out of if you're a mosquito. Right, very good. Right, that's all I've got time for. Next time we'll look into something which we're all familiar with, and that is eyes. Right? Okay, I'll see you next time.